All right, welcome back, everybody. Thanks for sitting through our two micro presentations this morning. We really focused a lot on non-sterile products this morning, and just for your information, this afternoon there's going to be more activity on the sterile products side. So any questions that you might have regarding sterile products, if you already sent them in, I would encourage you to actually resend them when we have our presenters on sterile products, just because it'll be more focused, and then John and I can spend our time talking about non-steriles. Also, we are part of FDA's Center for Drug Evaluation and Research, and so any biologic products, the recommendations that we have may not apply, and you will probably want to speak to the people that you interact with in CBER if you have any questions about those products particularly. All right, we have a number of excellent questions, and some of them are very similar. So what I've done is I've tried to go through and kind of group them together by subject, and so I'm going to try to, rather than calling out any one person's question, address them as a broad swath. So starting out, there were some questions about antimicrobial effectiveness testing. Specifically talking about when to test. So one question was, is USP 51 testing mandatory at beginning and end of accelerated and long-term stability for non-sterile drug products? And so that probably goes back to the slide that I was saying where antimicrobial effectiveness testing should be performed at the end of shelf life. And so what we do request is that one primary stability batch, so whatever your primary stability conditions are, one primary stability batch antimicrobial effectiveness testing is done at the end of that shelf life. So that's specifically what we're talking about. We're trying to decide if we're gonna.
Okay.
Yeah. So here's John.
Sorry, I was asleep at the switch here. Okay, so I have a question. And I'm going to read it in its entirety. We are in process of IND filing for a clinical trial. In a CMC section, we have to prove that the product is safe. For the clinical trial, we have to generate a certain amount of clinical grade samples. And the question is, if my process takes 10 steps and each step is passed by quality control, do you suggest validation should be done in last stage or every step in quality control testing? So that's a big question, a long question, and it does remind me that I did want to share with the audience that the Office of Pharmaceutical Quality has a mailbox for policy-related inquiries. So I want to share that with you now, and then I'll address the question. But for general questions regarding quality, you can send an email to CDER-OPQ-inquiries@fda.hhs.gov. Okay, so that's for future use. CDER-OPQ-inquiries@fda.hhs.gov. So this particular question gets to an IND filing and clinical trials and CMC. So my comments will be specific to microbiological quality and not the chemistry quality. I'll leave those for my chemistry colleagues. Again, when you send your questions to that OPQ policy inquiry mailbox, there'll be people that monitor that mailbox. They will farm those questions out to the appropriate subject matter experts and return a response to you. In terms of this particular question in microbiology for an IND, I will tell you that CDER does not want to inhibit drug development, and therefore during the IND stage, especially in the early stages, there's no requirement for validation of your sterilization processes. Okay, so that comes later when you submit your application for marketing. However, if I were doing a review for an IND as a microbiologist, what I would want to know is that if the drug is intended to be sterile, that you have sterilization step in there, that you test for sterility, and that you test for bacterial endotoxins. However, there's no requirement for those processes to be validated. That comes later in drug development. That's the end of my response.
So now I'll take one that's talking about BCC. And the question is, what suggestions do you have for performing a BCC risk assessment on a purified water system that is routinely monitored per USP criteria? I think this question was asked before John's talk started, but I think he had some really important notes in there about what went wrong with the water system that was in question at this time. It was poorly engineered to have dead legs in places where microorganisms could grow. I also note that the USP monograph for purified water doesn't have any kind of microbial count associated with it. It's just a total organic content specification. And so a total a microbial count might be something that you want to look into for a purified water system, as well as monitoring for the presence of any gram negatives or specifically Burkholderia. Those would probably be some good approaches to take. You want to make sure that a good routine cleaning schedule is implemented for a water system. That'll be important to make sure that BCC aren't able to establish there and grow those nasty biofilms that can be so difficult to get rid of. So those are some suggestions that we have for your water systems to keep the BCC out.
Okay, so I have a question here, and it says, could you please discuss the concerns that need to be addressed when Burkholderia is found in the process stream for a parenteral drug? So the question gets to, now we're talking about sterile drugs, and what should a firm be concerned about if they find Burkholderia in the water in that case. So I will start by saying, if the drug is intended to be sterile, as a review microbiologist here, I'm going to make sure that your sterilization process is validated such that it has an appropriate sterility assurance level. And if that's the case, then any Burkholderia that happen to be present in the water what goes into your drug solution prior to sterilization, those organisms would be killed. So that, in my mind, is not a patient risk. Now, at the same time, and I think Erika just touched on this, you do need to be concerned about upstream controls and things that may contribute to, for example, endotoxin load in your final product. Burkholderia is a gram-negative organism. You don't want to have a high endotoxins load even prior to sterilization, because we know that sterilization steps don't mitigate endotoxin. So those are my, off the top of my head, comments regarding concerns about Burkholderia in the water systems for a drug which is ultimately going to be manufactured as a sterile product.
All right. Okay. So, Erika, is the BCC risk mitigation strategy applicable only to aqueous, non-sterile drug product manufacturing? So that's an interesting question. So, yes and no. So while BCC are going to be at their most risky, essentially, for non-sterile products, because patients are going to be exposed to microorganisms in non-sterile products, you still want to have an idea, even for sterile products, of what kind of microorganisms are in your environment. So what's in your water system, what can contribute to endotoxins in your product, things like that. So while you may not be on the lookout as much for BCC in a sterile product, there are still concerns for what's in the environment for a sterile product. Does that make sense, John?
It does.
All right. Thanks.
As a quick aside, Erika and I joked about bringing noisemakers in for the Q&A session, but we stopped short of doing that.
We're going to have the Morning Zoo radio show.
Sorry for taking up some time to share that with you. But okay, so my next question, and I'm really glad that the questioner asked this, because this gives me a chance, again, to emphasize this very important point. The question is, do all oral solid dosage forms, are they required to test for Burkholderia? So the answer is no. And the longer answer, if you recall, Erika's presentation talked extensively or discussed extensively the issue of water activity and the importance of water activity regarding microbiological quality. So a dry product, although you certainly don't want to have huge numbers of microorganisms in a dry product, the difference between a dry product and one that's aqueous is that the dry product will not support the growth of microorganisms over that product's shelf life. So our concerns with Burkholderia, again, and this can't be underestimated in my opinion, our concerns at FDA with Burkholderia are with the aqueous non-steriles. Why non-sterile? Because as stated, the sterilization process will kill Burkholderia. Why aqueous? Because we know Burkholderia shows up in water systems, and if your product is aqueous, then it has a water activity high enough to allow for proliferation during shelf life. So numbers that come into that product initially that may be low during the shelf life, there's an opportunity there for those numbers to increase to an infectious dose size, which is dangerous for patients. All that being said, we don't feel that there's a need to test solid oral dosage forms for Burkholderia. Thank you for that question.
And I actually have a lot of questions on my sheet about water activity, so let's talk about that a little more. Some people were saying you can measure water activity theoretically, but what about in a practical setting? Well, water activity is actually something that is fairly easy to measure for drug products. You could purchase a water activity meter, and things that you can look up online and see what kinds of suppliers are out there for them in your area, and use that water activity meter to assess how your product, the water activity of your finished product, and whether or not that changes over its shelf life. This is definitely something that's fairly easy to address. So if you do end up having your water activity information about your product, and you think that that can help contribute to qualifying for a reduced testing schedule, such as a skip lot type testing schedule, that's the kind of information that you're going to want to include in your application. So it would be good to provide all of your developmental information in your application so that the reviewer can see, what your case is, what kind of great low water activity you have for your product, what your case is for asking for reduced testing, and something like that. So it's important to include all of that developmental work that you do so that people can see why your product should qualify for a reduced testing schedule.
Okay, my next question is long and has multi-parts to it, so I want to read the whole thing and then attempt to answer it. And the questioner writes, "It seems lucky that this firm conducted their surveillance with such diligence. How do we know that hundreds of other firms that are not so over and above have not formed biofilms within their water systems? Question mark. What are the implications of BCC for patient health from this product? How is surveillance for patient risk assessed? So this is an excellent set of questions. I want to begin by saying one of the reasons that we in the Division of Microbiology Assessment have looked at this as such an important case study to share with industry is the fact that the company, because they were going above and beyond, this is a success story in the end. This was a contamination case of drug, but it did not reach the level of patient infection cases, as some of them do. And all of you on the phone, you're aware of this from very recent news headlines involving microbiological quality and drugs and other types of products. So the good news here is that because the company was diligent, both, as I said, before the contamination and after, this did not reach the level of making news headlines with infections and patient adverse events. So that's the good news here. That's why we want to share this. Now, your other questions, the fact is that every company is different in terms of how they make a product. And, you know, Erika and I are involved in assessing quality. We're part of CDER's huge body of reviewers in Office of Pharmaceutical Quality. And this is the reason that FDA exists in terms of these questions. It says here, how do we know that hundreds of other firms that are not so over and above have not formed biofilms within their water systems? You know, as a healthcare consumer myself and having family members that consume healthcare products, I got a vested interest in these products just like you do. We all have a vested interest in making sure that the products that we take have appropriate quality. And I would offer that any of you on the phone, if you're going to make a major purchase on a car, you do your diligence in terms of which companies produce better quality products before you do that. However, when you sit down or put your daughter down on the doctor's table and the medicine is coming, you have no ability to evaluate the quality of that product on the spot, and you're taking it as faith that that product has good quality. And what is the reason behind that? The reason is that there is such a thing as FDA, and it's on us to do the best job that we can in terms of assessing how companies manufacture things and the resultant quality that comes with that. So the answer to this question, how do we know that hundreds of other firms that are not so over and above have not formed biofilms? Well, the answer is that the responsibility lies with the firm in marketing their product to make sure that it has good quality. And there is such a thing as FDA that is doing the best that we can to make sure that the firm is doing this the right way. The question continues, what are the implications of BCC for patient health from this product? Well, Erika's presentation highlighted some very recent events with the other products, not this particular case, where people were very sick. So we know that, we've seen it, and therefore these are the reasons that we have the concerns that we do regarding Burkholderia. How is surveillance for patient risk being assessed was the last question associated with this. And again, this comes down to FDA performing a pre-market review before a product is approved. And as was described in our sessions today, if you're a company that is going to send us an application for an aqueous non-sterile drug, we're going to make sure that you have a BCC mitigation policy in place. Related to that, another point that I did want to make, which I'll make now, is that if your manufacturing process is such that the Burkholderia can't live through that, then we wouldn't require you to test. If you send your bulk solution through a 0.45 micron filter, or if you have a heat step in the manufacture of your non-sterile drug, then there'd be no reason to have a Burkholderia test. But in the absence of those things, we're going to ask you to develop a Burkholderia release test before we're going to approve your product. Thank you for the question.
There really are a lot of ways to address that Burkholderia risk assessment. There was one question that sometimes products have a really low bioburden, and so if the firm doesn't have many colonies on their microbial enumeration plates from 61 testing, and if they were to send all of those out for typing by DNA or something like that, that could demonstrate that BCC are absent from the particular batches tested. There's really a lot of ways to approach that. My understanding is that USP has the BCC test, a compendial BCC test in the works. Is that your understanding, John?
It is, and we look forward to seeing that BCC test in the USP soon.
I think that's something that will take a lot of pressure off people for having to develop their own and validate their own tests. So speaking, John kind of has addressed one side of risk, of product risk, and talking about these aqueous products. We know that an aqueous product there's a lot of opportunity for microorganisms to proliferate in there, and so we consider those, of all the non-sterile products, we consider those to be the most risky. But what about on the other end, those dry formulations, those solid oral dosage forms, things like that? We had some questions about skip lot testing and what kinds of risk assessments to perform when pursuing a skip lot testing strategy for a very low risk product. And with that in mind, I'd like to direct you to ICH Q6A. There is a decision tree, I believe it's decision tree number 8 it's the last decision tree in the document itself, but it has a really good sort of flow chart as to how to decide whether or not skip lot testing is appropriate for your product, what kinds of information to look at your product for, and consider putting in your application when you get to that point. Skip lot testing is not going to be something that is acceptable for all non-sterile dosage forms, particularly because of the risks involved, but there are some that would qualify for it. We consider these things on a risk basis. We're not going to ask you to do testing that doesn't need to be done, but this is a situation where you definitely need to know the risks to your product and the risks to your process in order to make an informed decision and describe your informed decision to the reviewer. So.
Okay, thanks, Erika. My next question, if BCC is found in a diaphragm pump and the pump is replaced, is there still a risk of finding BCC in holding tanks and hoses that were connected to the pump? And I would say, I suppose there is that risk. I will preface my response with a statement that I am not a facility expert. There are many folks at FDA that are facility experts, and those folks are my colleagues that do inspections and are in your manufacturing facilities frequently. Off the top of my head, this question asks, does equipment that's downstream of a pump that was contaminated, do they run the risk of having BCC? And I would say yes, I would think so. I would think that you would want to be testing the water that comes out of that system to make sure that you have in fact mitigated the issue with BCC.
All right. So I'm going to go into some, I guess, not random, but just kind of disjoint questions about antimicrobial effectiveness testing in hopes of being able to address all of them in our Q&A period. One question is, if a product meets USP criteria for antimicrobial effectiveness testing, but not EP, European Pharmacopoeia criteria, is there any room for negotiation? So since we're U.S. regulators, the chapter that we typically look to for our antimicrobial effectiveness testing is the USP chapter. However, from time to time, we do have firms that manufacture product in Europe, and they perform the test as described in the European Pharmacopoeia. And that's something that we consider to be fine. These two tests are not harmonized. There are some differences between them, but they're not differences that are significant enough to necessitate redoing the test just to meet USP criteria. And that said, I'm not speaking for my European colleagues. That would be a question, if USP criteria were acceptable, that would be a question that you would want to direct to them. So.
Okay, I have a long question here, and I want to read the whole thing. And my reason for, in addition to addressing the questioner, is I want folks that are in the industry to hear this that may not be microbiologists, but may be in a position to be hiring them. So the question is, the industry for sure is evolving, but not enough microbiologists are in the product development phase. Do you see this lack of involvement from your perspective? And do you expect that microbiologists are on the staff for initial product development and test method development? I believe this could help with forward-thinking microbiological considerations and fewer issues once the product is on the market. Prospective versus retrospective approach. Tell me your thoughts. And questioner, I totally agree with you. We need more microbiology expertise in pharmaceutical companies right from the get-go. I know that, and this is something that I hear when I'm at conferences, that there is not enough microbiology expertise in companies, and so this movement to add microbiologists to the staff, I think, is a very important one. I will say that in the recent past, people with chemistry expertise have been making microbiology decisions, and I certainly have nothing against chemists. I was a chemistry minor myself as an undergraduate, but the fact is that chemists do not have training in microbiology, and these issues are things that you really need a microbiologist, some of these issues, to dig into and to understand and to help reach a suitable solution to. So yes, questioner, I agree with you, and it's important to add microbiology talent to your firm if you don't already have it. I think some of the submissions that we see, when we're reading them, we have a pretty good idea that the person that's writing the submission doesn't have a background in microbiology.
I could not agree more, and I definitely agree with this questioner too about the need to seek out and hire and develop microbiology talent in the field. You know, you wouldn't want a microbiologist making decisions about important chemical implications in your product. Well, why would you want to go the other way and have a chemist or an engineer, all intelligent individuals? Why would you want them to be making decisions about the microbiology aspects of your product?
I have a really good question here that came out of someone who was very astute and scrutinizing that figure that showed the red and the green boxes and when the biofilm was forming. So the questioner asks, if a biofilm was responsible for the contamination, how was a batch manufactured in between two contaminated batches that did not have a contamination issue? And I think that's a really good question. So again, to summarize, there were two red boxes in there with a green box in between. And I think the answer to this gets to the fact that we are dealing with microbiology, and this is an interesting and challenging science. My own feeling is that particular batch, for whatever reason, had lower numbers of organisms when it was manufactured, and the lower numbers did not register in that particular test that day. But that's something that we really will never know at this point. But one way of thinking about it is that the batches that came later and were consistently hot with the later testing, that was an indication that that biofilm was well established at that point and pumping high numbers of organisms into the drug solution.
So we're going to take one last question, and I just got a sheet, and I'm trying to kind of parse out the number one question on the sheet.
Do you want me to go while you're thinking?
Go, John, go.
I've got several here.
You got time for one more.
Okay,
Yep,
I'm
that's
told
fine.
by my handlers that I have time for one more question here. The question is, BCC seems to be a concern only for FDA. Has the FDA shared their health risk concerns with other global health regulators? And the answer is that, at least in my experience, I don't reach out specifically to other regulators. They're certainly aware of our concerns about this. I was just in Portugal during February to give this presentation, and there were 2 European regulators I know at that meeting who sat through this presentation. So that's my answer to that question.
John, isn't EMA in London? Maybe they can send us there. That's what we can do. So I think that's a good idea. All right. Well, thank you all
Wish
so
I had
much.
a noisemaker, Erika.
Thank you all so much for participating in the Q&A. And we've got Brenda here to give you some last parting notes for this morning session.